For decades, the standard timeline for mammalian domestication followed a strict logic. As human societies transitioned to Neolithic agriculture around 10,000 years ago, they selectively bred wild animals into docile, utilitarian variants. This timeline seemed to lock into place with the discovery of a 9,500-year-old human feline burial site on the island of Cyprus. An 8-month-old kitten, deliberately interred alongside human remains, appeared to be the definitive proof of early domestication. But osteological evidence, bone fragments, and skeletal structures, creates a blind spot. A wild animal that is simply tamed during its lifetime leaves behind physical markers identical to an animal that has undergone genuine genetic domestication. When paleogeneticists extracted and sequenced ancient DNA from hundreds of these early feline remains, the genetic data contradicted the accepted agricultural model entirely. By synthesizing modern paleogenetic data and tracking mitochondrial lineages across thousands of years, we can map a much more complex two-wave biological history of Felis catus. Equating a commensal relationship, where animals simply live alongside humans for mutual benefit, with actual domestication, obscured an evolutionary timeline that operates completely outside the rules of human engineering. To see the biological distinction between physical proximity and domestication, we examine the agricultural settlements of the Yangshao culture in ancient China, dating back 5,400 years. Archaeologists found small cat bones scattered throughout these early farming communities, but this animal, with its distinctly spotted coat and unique facial structure, was not an early version of the house cat. Mitochondrial DNA extracted from 22 ancient specimens identified these remains as leopard cats, Prionolurus bengalensis, a separate evolutionary branch divorced from modern domestic breeds. For 3 millennia, these leopard cats exploited the exact same ecological niche that modern cats do. They patrolled grain stores, they hunted rodents, and accepted shelter in human settlements. Yet, despite 3,000 years of continuous interaction with human farmers, this relationship never triggered genetic domestication. The lineage died out as an evolutionary dead end. This localized failure in Asia stands in sharp contrast to the wildcat lineage originating in the Near East, which ultimately survived and populated the globe. Extended proximity to human agriculture does not inevitably yield a domestic animal. The eventual global conquest of Felis catus requires a specific genomic explanation. Extensive genetic sequencing isolates a single subspecies, the African wildcat, Felis sylvestris libica, as the sole wild ancestor to every modern domestic cat. Researchers track mitochondrial DNA clades inherited along the maternal line. By mapping these genetic markers from ancient remains, we can reverse engineer how this specific subspecies moved across the continents. This animated map tracks the expansion of specific feline genomes across the Old World, beginning around 8000 BC. The first major genomic wave consists of mitochondrial clade 4A star. This lineage originates in Anatolia and diffuses westward, reaching Southeast Europe by 4400 BC. A geographic split prior to the formation of the Black Sea isolated these groups, causing minor genetic drift between the Anatolian and European clades. This expansion was a purely natural migration. These wildcats simply followed the swelling populations of the house mouse, Mus musculus, which were flourishing around early human grain silos. The initial wave of feline expansion was an act of ecological opportunism, driven entirely by food supply, without any human-directed selective breeding. A second distinct mitochondrial lineage eventually emerged further south, specifically within North Africa, clade 4C. The primary paleogenetic data for this lineage was extracted from Egyptian cat mummies, dating from the 7th century BC to the 4th century AD. Local Egyptian decrees strictly banned the export of cats. Genetic data shows those laws failed. Returning to the map, between the 5th and 13th centuries AD, clade 4C erupts out of Egypt, spreading aggressively across the Mediterranean basin and deep into northern Europe. The primary vector was maritime trade. Seafarers kept cats on board to combat the black rat. This Egyptian lineage abruptly appears in distant locations, from Roman ports to the Viking hub of Ralsvik and along the Silk Road. By the Tang Dynasty, roughly 1,000 years ago, this Egyptian lineage reached China, completely outcompeting the native leopard cats that had occupied those settlements. Thriving in the confined, high-stress environment of ancient cargo ships required a level of sociability absent in the solitary wildcats of Wave 1. A specific behavioral shift had occurred within this Egyptian lineage. We must ask what biological mechanism allowed this specific lineage to tolerate cramped human ships, while other feline populations remained strictly solitary. Paleogeneticists isolate behavioral mutations through comparative genomic analyses, contrasting the DNA of ancient wildcats against their modern domestic descendants. The findings align with the neural crest hypothesis. This biological model dictates that minor alterations in early embryonic stem cells can rewrite the development of a mammal's central nervous system. The genomes of the Egyptian lineage contain overlapping mutations in the genes responsible for both dopamine production and fear conditioning memory. These specific mutations amplified reward-seeking behaviors. The cats began to experience a distinct neurological dopamine release in response to human interaction, such as receiving food treats or being stroked. This dopamine-driven reward loop manifests physically through enhanced pupillary and auditory responses triggered by human interaction. Other large African felines, such as lions or cheetahs, lack these precise dopamine-regulating mutations. Their nervous systems retain the deep-seated fear responses necessary for survival in the wild. These invisible neurological mutations provided a critical evolutionary advantage. By predisposing the Egyptian lineage to tameness and sociability, the cats were neurologically equipped to survive and disperse within human-dominated spaces. In mammalian biology, behavioral tameness is almost immediately accompanied by morphological changes, a dynamic known as the domestication syndrome. In species like dogs or pigs, this manifests quickly as floppy ears, shorter snouts, or highly variable coat colors. Yet Felis catus presents a distinct biological paradox. Despite their global expansion and behavioral tameness, cats lacked these physical markers of domestication for 9,000 years. This diagram maps the TOCPEP gene, a molecular marker for physical selection. The dominant TAM allele produces ancient mackerel stripes. A specific genetic break, the W841X mutation, is required to activate the recessive TAB allele, generating the blotched tabby pattern. Sequencing data from ancient feline remains reveals that this W841X blotched mutation was virtually non-existent in the population for thousands of years. We see the opposite pattern in species like horses, where human breeders actively selected for diverse coat colors at the very onset of their domestication. The total absence of aesthetic mutations proves that ancient human societies were not actively breeding cats. They utilized the animals strictly as functional tools, not as engineered pets. Historical iconography corroborates this genetic data. Egyptian and medieval artists consistently depicted their cats bearing the vertical stripes of the wild-type genome. The recessive blotched allele only achieves a high frequency in the global feline genome well into recorded history. This genetic shift maps to the Middle Ages and the Ottoman Empire, centuries after the Egyptian lineage had already established itself across the globe. The aggressive explosion of diverse feline phenotypes, the vast variations in size, coat length, and facial structure, does not truly begin until the rise of fancy breeding in the 18th and 19th centuries. For the first 9,000 years of their relationship with humans, cats executed a process of self-directed behavioral adaptation and purely opportunistic geographical expansion. Deliberate, human-directed selective breeding occupies only the absolute final fraction of their evolutionary timeline. Felis catus effectively domesticated itself. It modified its own neurological pathways to exploit global human infrastructure, securing its survival millennia before humans ever attempted to control its genetic code.